My name is Maria Cuervo, and I'm interested in the study of autophagy. That is something that happens in each of your cells every day, and it's a process that eliminates any kind of damage or garbage that accumulates inside your cells. We found a while ago that autophagy decreases with age, and we think this contributes to the building up of garbage inside the cells as we get old. And there is evidence not only from our groups, but many other groups now showing that this failure of autophagy with age can be a very important aggravating factor in many diseases. The ones that we are particularly interested are three categories. One of them is neurodegenerative diseases such as Alzheimer's or Parkinson's, where the main affected organ is your brain. The second major group is metabolic disorders, and you also know about diabetes, obesity, cardiovascular conditions. And the third block of diseases is cancer, because aging is the biggest or the common risk factor for all types of cancers. The main goal of this research is trying to understand why people who have Alzheimer's disease have also higher risk of having other conditions. So, for example, there are these studies showing a connection between having Alzheimer's and having diabetes, but the mechanism is not clear why this happened, and we think that failures in autophagy might provide an explanation for this relation. The new grant that we got is actually a collaborative project among 6 groups at Einstein and 2 in other institutions, and it's the continuation of 10 years of research where we've been studying the changes in autophagy with age, and now we are moving to a new period on this grant, and we actually need all that expertise to study the individual as a whole, not just isolated organs. So my group is interested in the brain, and we will be contributing information on the brain functioning and autophagy. We have Fernando Macian from pathology, and his group will be contributing immune response, mostly tcells. Rajat Singh, that is our major metabolist expert from medicine. We have Britta Will, that is the expert on stem cells at very different levels, but will focus in the immune and hematopoietic system. And then Ebris Gabathiotis, that very known for his development of drugs in other areas, and that we've been lucky to recruit to work in autophagy now and develop drugs for modulation of autophagy, as well as Mimi Kim, who is bringing all this data together with her biostatistic power. And then outside Einstein, we have Laura Zantambrosio, currently at Cornell, and she started at Einstein and has traditionally been looking at lympha or how the brain and other organs communicate. And then we have a major expert in imaging, Jose Javier Bravo Cordero. He did his postdoc at Einstein and moved to Mount Sinai, and he has expertise in very sophisticated image processes that are required to study autophagy. So if autophagy doesn't work, not only do you accumulate garbage, but you also accumulate fat. So for example, doing studies both in humans, but also in mice, I'm gonna show you what happens when you don't have autophagy in your brains. You can see in white is the profile of different cells in your brain. The blue corresponds to the nucleus, and then those green dots that you see there, they shouldn't be there. This is fat that is accumulated inside the cells in the brain. And the only thing that we have done to these animals is to eliminate autophagy. So this suggests that having functional autophagy is really preventing normally this accumulation of abnormal fat that is going to compromise the way that the brain cells function. We think that these results with this accumulation of fat in the brain of our animal models is very relevant to disease, because in collaboration with Randy Nixon in NYU, we found that if we analyze the brains of patients that had Alzheimer's disease and we look at their cells inside the brain, we can see similar accumulations of fat, and we don't see that in people over the same age if they don't have the disease. We found that when proteins cannot be eliminated in the brain because autophagy is not properly functioned, they go to the periphery, they go to the rest of the organs of your body, and they can compromise the function of these organs and contribute to other diseases. So for example, this is the liver of a mouse, in which in the left you see a healthy mouse, and in blue is the nucleus of these cells in the liver. And as you can see, there is nothing green around. But if you compare on the right, this is the liver of a mouse model of Alzheimer's disease, and the bright green corresponds to tau. So tau is a protein that is present in the brain, and that in this case we found it located in the liver. It's a place that it shouldn't be there. And we know that in part, or we propose to investigate, that the failure of autophagy in the brain is dumping these proteins to other organs. So we have seen these proteins reaching the liver, the pancreas, the adipose tissue, so many organs, and we think that this will contribute to loss of function of these organs and could also make them much more vulnerable to disease. Centenarians, that as you know live longer and healthier, they seem to have much better autophagy. One of the findings that we were particularly excited is the discovery that these changes with age that we see in autophagy in animals also happen in people. And this is actually work from Fernando Macian. So he collaborated with Nir Barzilai, trying to understand whether centenarians, that as you know is people who live more than 100 years, were having better autophagy. T cells are cells that are in your blood, and some of these T cells remain with you per life. So as you can see in this image, these are T cells from people that came from a centenarian family on the top, or non-centenarians, but the same age in both cases. In blue you can see the center of the cell, the nucleus, and what you see in green are spots or places where autophagy is happening. And as you see in the top, you have very active autophagy in all the cells, in contrast with the low autophagy on the bottom ones. So this is extremely encouraging, because we want to make everybody centenarian regarding their autophagy and increase their autophagy so they can fight all these toxic products and all this garbage that accumulate with age. So if we are able to at least restore autophagy at the levels that a centenarian person has in the general population, we might be able to fight part of this damage that accumulate, and this might slow down disease or prevent some of the diseases of aging.